Thank you for joining us for this webinar to look at the new Triton-free master mixes and chemistries. My name is Dusty Vyas and I'm part of the Technical and Application Support team at LGC Biosearch Technologies. Today we will cover the reasons that the formulations needed to change. I'll explain which of the LGC chemistries required the reformulation. I'll talk you through the extensive testing that was carried out on different tissue types, extraction methods, platforms and assays. And finally, I'll show you the data generated to illustrate the equivalent performance. REACH was established by the European Chemical Agency, and its remit is to assess the production, import and use of chemical substances and their potential impacts on human health and the environment. In November 2017, several classes of chemicals were added to the REACH regulations, which prohibit the use, production and sale within the EU. One chemical is the detergent Igipal CA630, also known by the trade name Triton X100. By January 2021, sales of products containing Triton X100 will be prohibited within the EU. It is therefore necessary to reformulate several products to be Triton-free. So let me take you through which of our products are changing. All of our KASP Master mixes have been reformulated to include an alternative detergent and now are Triton-free. We have taken this opportunity to streamline several of our KASP legacy products to improve efficiency and simplify the ordering process for our customers. The table on the right details the legacy KASP Master Mix formulations and the equivalent Triton-free formulations to be used following release. Some of our other products in our portfolio also contain Triton, so these have also been reformulated, and these include the Replicator Wash, Clear Jeans and Mag Beans. These will not be part of this presentation. Information on these can be found in the accompanying PDF. We have carried out extensive testing and validation of the Triton-free formulations, and we have tested every scenario we could think of to ensure that the data generated is not affected. This included over 850 PCR plates and the generation of 580,000 data points. Robust testing involved testing a range of tissue types, extraction methods, platforms and assays. And we want to emphasize that we have seen equivalent performances of the new Triton-free formulations when compared to the legacy master mixes. A wide range of tissue types were incorporated into the validation process, and to ensure the Triton-free mix demonstrated equivalent performances for all sample types. This included seeds, which were maize, wheat and tomato. These were chosen as they are popular customer crops and are genotyped frequently. Sunflower seeds were included as the seeds are oily and contain secondary metabolite content and enabled the new mix to be tested for inhibitor tolerance. Human blood was also used, as we have many well characterized assays, and these can be used to identify subtle performance differences. Lettuce leaf was the representative leaf tissue used, and in total we extracted DNA from over 1,000 plant samples. Another criteria that was included in the validation process was to look at the extraction chemistries. A wide range of extraction chemistries were included in the validation. These included 3 Biosearch technology extraction and purification chemistries. There was SpeedX, clear gene, quick extract DNA extraction, and a crude extraction method that was included was the sodium hydroxide hotshot DNA extraction. And this was to encompass all the possible chemistries that our customers may use as part of their genotyping process flow. The validation was performed in both 384 well and 1536 well plates on our SNP-line platform. We also ran validation on the Nexar platform in an array tape format. A wide selection of assays were used and we had at least 8 per species. We also made sure that assays requiring non-standard cycling conditions were also included. Different cycling conditions are required for some assays due to the melting temperature of the primers and whether the region is AT rich. The percentage GC range from 21 to 72%, encompassing a wide variation in the GC content. In order to validate the new formulation, it had to meet a range of criteria. These were a qualitative inspection, which was carried out by 2 senior scientists, quantitative analysis of NTC performance, call rate and data concordance was performed for every data set. So let me take you through some of the data that was generated. In this plot we have leaf tissue from lettuce, which was extracted using quick extract and genotyped in a 1536 well format using standard cycling conditions. We achieved a call rate of 98.5% and concordance was 100% between the 2 mixes. The fact that they cluster so nicely illustrates how robust our mix manufacture and QC processes are. This plot is an example of sunflower seeds extracted and purified using SpeedX extraction chemistries and genotyped on the array tape using standard cycling conditions. We achieved a call rate of 96.8% and concordance was 100% between the 2 mixes. This plot is an example of human blood extracted and purified using clear gene chemistry and genotyped in a 3/8 well format. Using optimized cycling conditions, we achieved a call rate of 98.3% and concordance was 100% between the 2 mixes. This slide shows an example of the range of completed validation experiments. It covers the full range of sample types and DNA extraction chemistries, and the number of plates that passed each of the criteria. Underneath in brackets is the number of plates required to meet our validation criteria. So to summarise, LGC Biosearch Technologies has had to make essential changes to the formulation of products that contain the detergent Triton to comply with the new regulations. This included the whole KASP Master Mix portfolio, and to ensure that this change had no impact on our customers, we performed extensive validation of the new Triton-free mix against the current formulations. This included a representative range of species and tissue types, over 850 reaction plates, and the generation of over 580,000 data points were analysed as part of the validation. So the change to the Triton-free formulation has no impact on data generated. And if you have any further questions, please contact us at the email in this slide. I'd like to take this opportunity to say thank you to everybody for your patience and time.